So I'll be talking about gndrv, the biological data manager. This is what a computer program looks like right before it gets fed into the machine. And this is that same program while the programmer is actually working on it. Now this is a digital representation of what DNA looks like. And this is also what it looks like when someone's designing it. So this is extremely confusing, and as you might imagine, is ripe to produce little undetectable errors. Now these errors can set a scientist back weeks or even months in their research. So there's a huge need for new tools for this DNA development process. In particular, a linter or spell check for DNA. Now you might be wondering, why do we even care about designing DNA sequences? And there are a number of industries that do, but the biggest is synthetic biology. So synthetic biology is a field where scientists actually design DNA sequences and add them to organisms in order to make them do something completely new. It has applications in agriculture, energy, medicine, the environment, and more. It's also exploding. So 2016 marked the first year where there was over $1 billion in funding for synthetic biology companies. Additionally, with projects such as Bill Gates' Target Malaria, there is increasing excitement about synthetic biology's power to completely eradicate certain diseases. Now, when scientists go to design their sequences, there are a few solutions they turn to. And these solutions are made by very smart people, researchers in molecular biology. Unfortunately, they're confusing, extremely expensive, sometimes over $15,000 a year, and often insecure. So it turns out that while researchers are excellent at doing science, they just don't have the background to develop commercial software. So we should be turning to the people who do, namely the well-established software development industry. By taking inspiration from software development practices, gndrv aims to make the process of designing DNA sequences faster, easier, and more reliable than existing solutions. Currently on the website, gndrv.com, people can create user accounts, create parts, and see which parts other users have created. But going forward, gndrv will integrate a DNA verification process where scientists can enter a DNA sequence and check for common problems before ever putting it into a cell. Now, public repositories on gndrv are free, whereas private repositories will require a premium subscription, which will also come with things like unlimited verifications. There are over 6,000 students each year who participate in the iGEM synthetic biology competition, so this is a key demographic to target. Additionally, Benchling, one of the services I showed earlier, has over 150,000 users. Now this is my 4th year doing the iGEM competition. It's also my 2nd year leading Waterloo's math team. On previous co-ops, I've worked in both front-end and back-end web development, as well as bioinformatics, so I'm uniquely positioned to solve this problem. Now, the 5K will be instrumental in acquiring the talent and resources to accelerate development, to have a prototype ready for the iGEM Jamboree, and a full launch in February. Thank you.